All right, AP Bio, here is going to be the webcast for enzyme structure and function. So we'll kind of get like this will be the second of probably 4 or 5 of these. So when we talk about structure and function, structure always determines function, and so molecular interactions lead to organisms' phenotypes. And so when we talk about molecular interactions lead to organisms, those are your phenotypes, and so the genotype determines the phenotype. So metabolism is controlled by enzymes, which we're going to talk about. So again, this is a metabolic pathway. And so in this metabolic pathway, we have a series of enzymatic reactions here. And so this pathway, the whole point of this is to break down these interactions into smaller chunks, so to speak. So enzymes are mostly proteins, and so enzymes are molecules that serve as a biological catalyst, and some RNA molecules also have catalytic properties, which we'll talk about. But again, here's our primary structure, proteins, right? You get your primary, secondary, quaternary, tertiary. So enzymes are also proteins, which means they can also be denatured, which we'll talk about in lab. So again, I have another thing I need to rotate here, so this is going to screw up my whole presentation. So let me drag that over. All right, so when we talk about what enzymes do, they essentially, if we look at the red, that's the course of the reaction with the enzyme, and as we compare that to the black, which is the course of the reaction without the enzyme, the key point here is over here. The activation energy is lower with the enzyme, and so what I would strongly want you to understand is that by using enzymes, we save free energy. That free energy can be used to do work later. So the whole point is that those enzymes, they can lower that activation energy, and so they make the system more efficient. So it's all about energy. So that's the key point with that one. So as a catalyst, it's any substance that increases the rate of a chemical reaction by lowering the activation energy. I would underline that, I'd highlight that, without participating in the reaction. So the catalyst, right, in this case, if we look again, we just seen that reaction coordination. It increases the rate by lowering the activation energy, but it does not participate in the reaction, and so it's not going to change anything there. So again, with the enzyme, there's your activation energy, and so this looks like it's carbon dioxide and water, and you got some new products there. So we're going to label this picture in class. So those of you watching this, you may just want to redraw this in your bill. You can pause it here, and then pause it here to remove my face. But again, you've got your substrates enter the active site. So the substrates there are the orange and the green. The substrates are held in the active site by weak interactions, and then this is called the enzyme substrate complex. Then the enzyme substrate complex, a series of reactions will occur, and now you have new substrates have been converted to new products. So we have one color up here to a different color up here. We've got new products, and then the enzyme, the products are released. But as you can see, the enzyme has not changed. It is not. It's just been the catalyst, but it's not been changed during the reaction. And so enzymes work by physically positioning reactants to substrate in ways that increase the likelihood of chemical bonds being broken or formed. So enzymes are highly specific for substances that they interact with, and the name of the enzyme tells about the substrate. So ACE, so lipase would break down lipids. Helicase breaks up the DNA helix, things of that nature. So again, I just went through that whole catalytic process. So the induced fit model, when we're talking about induced fit model, that's how the enzyme interacts with the substrate. And so substrate molecules, they physically bind at the active site, and the binding causes a conformational change, catalyzes the reaction. And so the active site is probably the most important part. Excuse me, but that induced fit model, enzyme fits into, or excuse me, the substrate fits in that active site and the reaction can occur. So cofactors and coenzymes, these are substances that allow enzymes to work better. And so many enzymes require an organic, which are called coenzymes, or vitamins. So vitamin K, vitamin B, vitamin C, vitamin D, those are coenzymes. And so these are organic, so they've got carbon, hydrogen, oxygen. Cofactors are minerals, and these atoms, groups of atoms to be complex with the enzyme. So cofactors, if you think of like people that are anemic, so they need iron. So iron would be a cofactor. And so if you have a deficiency in coenzymes or cofactors, this could lead to enzymes not working properly, which can lead to metabolic issues. So many enzymes involved in interacting with DNA require zinc ions as a cofactor. And so if you look over here, those little green dots, those green spheres, those are a very important cofactor. So let's talk about regulation of enzyme activity. I'm going to try to keep this one maybe a little bit longer than my 6 minutes. But so other molecules can affect enzyme structure and function. So if we look at this top row, we've got competitive inhibition. So here's your enzyme, there's your substrate, there's the reaction. Again, active site, you have your enzyme releases that product. So that's your competitive. So in competitive inhibition, right? The top one's the normal reaction. The bottom one here, you have inhibition. And so in inhibition, this inhibitor, right, it's going to bind to that active site. When it binds that active site, it essentially blocks it. And so when that active site is blocked, this normal substrate cannot attach. And so essentially, you inhibit the enzyme from doing its job. So competitive inhibition, which again we'll talk about later, is this idea that by inhibiting the active site by blocking it, the substrate cannot be acted upon. Allosteric interaction is a little more complicated. So there's our normal reaction up top, right? It affects enzyme by binding away. That's the key, away from the active site. So there's our substrate enzyme. You've got your substrate molecule binding to it. You have your new products. In allosteric interaction, you have this space down here. So there's our inhibitor, there's our enzyme, there's our substrate. That inhibitor attaches to what we call the allosteric site. And by attaching to that allosteric site, we've now changed the shape of the active site. And so when that substrate binds in, it does not fit. Change the structure, change the function. So allosteric interaction allows for feedback inhibition. So for example, ATP inhibits the activity of phosphofructokinase, which is an enzyme important for pathways, which we'll talk about. And so ATP actually acts as an inhibitor, allosteric one. So we're going to be labeling these pictures in class. Those of you that are doing this online, you may just want to pause the video and draw these on your own, but we've got A, normal binding, B is competitive inhibition. The key there, you're blocking the active site, and C, non-competitive. The key part here is down at the bottom. You have that non-competitive inhibitor attaching to the allosteric site. So if you don't remember what an allosteric site is, you need to go back a couple slides. So again, we're going to be doing this one in class. My face is over the arrow. But what you need to do here is your 3,9 is an active site, and this is a metabolic pathway showing you these intermediates, enzyme 2, intermediate, enzyme 3, intermediate C, enzyme 4. And so these are a series of, these intermediates are these chemicals that don't last very long. And so the enzyme will act upon these molecules, but ultimately that's your end product, isoleucine. That isoleucine will then act like an allosteric inhibitor, binding to the enzyme, acting as a feedback inhibition. And so once you've produced this, the cell's not going to produce any more than it needs. So once it has a high enough level, it will stop this process. And then environmental influences, which we'll be doing in lab. So temperature and pH, they denature the protein. And so this is kind of that key. We already talked about denaturing, change of structure, change of function. So temperature and pH can affect enzyme function because of the denaturing of the protein. You change the structure of the enzyme, you change its function. And so concentration will affect it. We're gonna do this in lab. So concentration can affect it. And so when we talk about how to measure enzyme activity, which we're going to be doing in lab, often we're looking for a product. And in this case, we're going to be looking for the production of oxygen, because oxygen bubbles will produce and it'll cause those to raise. So, all right. That is all the enzyme stuff. It's 8 minutes. That's way too long for you to sit and watch, but it's the best I could do. Again, go through, watch AP dailies, do all that practice stuff, and reach out if you got any questions. This is me signing out from Room 102. Have a great day, and I'll see you sometime this week.